Good morning, everybody. We're just hitting 10 o'clock now. We've still got a few people joining, so we'll just give it a second and then we'll launch into our June PathSafe webinar. Okay, the numbers joining are steadying now. So first of all, welcome everyone to the fourth instalment of the PathSafe webinar series. Today we're joined by colleagues from FERA Science Limited who are leading the PathSafe Workstream 3A project, which aims to identify and assess portable diagnostic technologies that have the potential to be applied to foodborne pathogens. We'll start the session today with a brief introduction to the context of this project given by Rick Mumford, our Deputy Director for Science, Evidence and Research and Deputy Chief Scientific Advisor here at the FSA. The FERA team will then present an overview of the work that they have undertaken to date and plans for the next stages of the project. Following the presentation, there'll be a question and answer session. So firstly, just a few housekeeping elements before we begin. The session today has been recorded and the recording and the slides will be saved to the PathSafe website in due course. The session is going to be an hour long, running through till 11 AM. Could we ask that you please keep your microphone on mute when you're not speaking? And as mentioned, we will have a question and answer session following the presentation. Please use the question and answer function to post any questions you have. And when we come to the question and answer session, we'll ask you to unmute and ask your question. Any questions that we don't get to or can't be answered online, we'll follow up offline after the meeting. And please note the chat function has been disabled for this webinar format. A link to the PathSafe webpage will be pinned in the Q&A section shortly for your reference. So with that, I will hand over to Rick to start us off.
Hi, Rachel. Thank you for that. So I thought it was just be interesting to give a couple of minutes just explaining some of the context and some of the background to Workstream 3 and explain why would we be interested in using sort of portable remote diagnostic technologies. So going right back to the beginning of PathSafe when the bid was put into the Shared Outcomes Fund, the whole premise was to look at new technologies, in particular new technologies based on genomics, so DNA technology. And obviously we're all a lot more familiar with the power of that since COVID, because many of us would have had a real-time PCR test and we all heard about the stories about wastewater testing, etc. So there's real sort of power in this technology, and PathSafe is looking at that in many aspects. So it's looking at things like whole genome sequencing in Workstream 1 and can we better characterize pathogens and antimicrobial resistance. In Workstream 2 is looking at things like wastewater testing, and can we look at a population level or an environment level where foodborne disease might be. But Workstream 3 is specifically focused on these portable technologies. So the idea, could you take something like real-time PCR out into the field and use it rather than a traditional, often slower approach whereby you might take a sample, send it to a laboratory who then test it and send back the results. So that's the kind of broad context to what we're doing here with this work stream. So it's all about pilots. It's all about looking at the feasibility of these technologies. It's not necessarily about, oh, we will do a test and then we'll roll these things out on April 2024. It's about, can these things actually work? We know there are technologies out there, but how close are they to being used in the field and put into the hands of professionals who can use them? So that's the kind of background to where we are, and this is giving us an update on the work that FERA have been doing to investigate that, put a framework around the different diagnostic technologies around there, and then see if there are particular pilots that we might be able to roll out. So I'm going to stop there. Hopefully that was good enough to set the context, and I think I'm going to hand over to our colleagues at FERA now. Is that right, Rachel?
Yeah, that's correct. Thanks, Rick.
Okay, thanks.
Hi everyone, can everyone hear me and see the slides okay?
Yeah, that looks great. Thanks, Ashley.
Okay, great. So good morning, everyone. Today we're going to be talking about PathSafe Workstream 3A, which is a horizon scanning and technology readiness level study with in-field testing of rapid diagnostic technologies. So today we're going to be discussing what we've done so far and then our next steps. So here's just a picture of our team for the project, made up of scientists from FERA and then also a collaborator from the University of Lincoln. And today I'm going to be talking about the literature review and evaluation of technologies that we've done during the project. And then Barbara's going to be talking through some of our engagement with stakeholders and end users. So the work we're undertaking is separated into 3 work packages. So the first is centred around a literature review of the technologies available for on-site testing, either currently applied for foodborne pathogens, but also a broader search to include technologies applied in other fields such as plant health, veterinary sciences, or human health. And alongside this, the development of a technology readiness level framework to evaluate where these technologies are in their development and readiness for deployment. So traditional frameworks remain heavily based on sort of NASA descriptions, but the aim was to modify this framework to make it more appropriate for our work. And then finally, putting these together to create a database of the identified technologies and their assessed technology readiness levels. Alongside this, work package 2 involved engagement with stakeholders to look at where we could make the biggest impact, where technologies could be best deployed and what benefits they could provide. And then the results of these first 2 work packages, we're going to go into work package 3, where we're going to pick 2 technologies to take forward and develop in pilot studies to try and increase their readiness for deployment. So here's just a demonstration of the scope of our project. So we were focusing on foodborne pathogen targets such as norovirus, Campylobacter, Salmonella, Listeria, Clostridium, and other indicator organisms such as E. coli. And the sample matrices included were water, meat, shellfish, dairy, swabs, animal feed, fish, fresh produce, and ready-to-eat products. So quite a broad scope. So we had 2 searches in our literature review, one which focused on on-site methods for foodborne pathogens, and then a more general search which was looking at on-site novel methods in any field. And our literature search looked at literature from the last 5 years, so from 2018 to 2023. So these searches returned about 28,000 papers, 8,000 which were removed by the software we were using as duplicates, but we then had around 16,000 papers to screen by reading the title and abstract and deciding whether it was relevant and we wanted to take it forward to our full text review stage. So we then had over 3,000 papers to evaluate in the full text review, and these were grouped into the technologies which the paper was looking at. So at the end of our initial screening stage, we had a table which looked something like this. This is just a small part of it, but you can see the results of the technologies we found during the literature search and then the numbers of papers in each category, to sort of give us an idea of how prevalent they were in the literature. So we then went on to evaluate each of these technologies using the full text literature according to our technology readiness level assessment. So now I'm just going to briefly go over our technology readiness level framework in a bit more detail. So we didn't really consider technology readiness levels 1 and 2, which in the NASA definitions are sort of around concept generation. And we didn't think these low TRLs would be very represented in the published literature or wouldn't be within the scope of the project to take forward in pilot studies. So the framework starts around technology readiness level 3, where laboratory demonstration of the technology starts, including experiments around the concept feasibility. Then technology readiness level 4 is when the technology is being applied to detection of a model pathogen. So this doesn't have to be an in-scope pathogen, but it's just been demonstrated with any target pathogen. And at this point, we're just talking about individual component technologies being tested within the lab still. Technology readiness level 5, the technology we're evaluating will have been applied to a relevant in-scope pathogen. And technology readiness level 6 is where the technology has been put together with all the necessary components which would make it suitable for on-site testing and its performance evaluated in the lab. So for example, when we're looking at evaluating these sort of molecular biology technologies, it would have had to be paired with a suitable DNA extraction suitable for on-site testing to reach technology readiness level 6. Technology readiness level 7 is when the technology has been tested outside of the lab in a relevant setting with end users. And to reach technology readiness level 8, it will have had its performance fully evaluated in an in-field setting. And then finally, technology readiness level 9 is when it's overcome any additional logistical barriers or hurdles and it's fully deployed or deployable. So we used a technology readiness level assignment tool to answer questions based on our framework about each of the technologies we were assessing to guide the technology readiness level assignment. And our assignment is just based on the needs of this particular project, using obviously the framework developed for the project and just based on the results of our literature search, which covered the last 5 years. So we ended up with a database with a summary like in the picture here, which again is just a small part of the overall table, where we had a number for the highest technology readiness level we thought had been achieved in any organism. So for example, if it had been deployed in a different area, for example, COVID-19 testing, then that would be shown here. And then we assessed the highest TRL achieved for each of the target pathogens. So we could see if it had been not applied to the target at all, it would be TRL 4, or what level it had achieved. So if it had been applied to a target but not taken outside the lab, or if it had been fully deployed in some areas for the target pathogen. So this gives us an idea if we're going to select a certain technology for a certain application, what the next steps would be in terms of trying to progress it through the technology readiness levels towards deployment. So now I'm going to hand over to Barbara to talk about the work in work package 2.
Thank you. Okay, yes, right. So as we saw at the beginning, I'm just talking and assume anyone or everyone can hear me and shout if you can't. But as you saw at the beginning of the presentation, the aim of Work Package 2 was to work alongside the science team and to help identify and integrate end user needs. So this way we wanted to ensure that the selected technologies are able to address, first of all, real world needs, and second, that we take into account any specific requirements to make them fit for purpose and to allow them to be integrated into existing processes. So we started by creating a map of all the different stakeholders based on the scope of the project, which again you saw earlier. And this is the result. So don't worry too much about the detail. I know the writing is very small, but the key message here is that the broad scope in terms of pathogens obviously also translated into an extensive stakeholder map. So we have domestic stakeholders across multiple primary industries and then also downstream along the supply chain. And in addition, there's obviously also monitoring and testing processes, including, for example, import controls. Yeah, so what we then did was on the next slide is we wanted to start with a more strategic overview of where gaps and needs may potentially lie within that supply chain and testing environment. And to be honest, also see if there's anything that we can possibly exclude straight away. But yeah, so anyway, so we organized a couple of focus groups with members from different organizations, including obviously the FSA, but also DEFRA, APHA, CFAS, and UK HSA. And then we also had a couple of individual conversations where people couldn't make the focus group. And then we followed those up with individual interviews with what we called operational end users. So those who would actually be either doing the testing themselves or have quality assurance teams to do the testing. And initially we were also planning to do focus groups here, but all the lovely bank holidays have made that a bit more difficult. So we ended up with one-on-one calls and conversations, or sometimes one-on-two if there were a couple of representatives from different teams in an organization that wanted to join. And yeah, there's still some last ones trickling in, but essentially we've covered a number of primary industries as well as businesses along the supply chain, including sort of processing, retail, up to ready-to-eat products, and also port health and laboratory testing. So hopefully we should have a good picture by now. And yeah, so all these conversations were really interesting. For me, lots of new information because I usually work in plant health. So on my last slide, I just wanted to briefly summarize what we spoke about, though I won't again go into too much detail. Unsurprisingly, there was quite a difference in perceived needs between statutory testing and non-statutory or additional testing. Essentially, most people were immediately gravitating towards additional testing as an opportunity for portable technologies because statutory testing by its very nature is obviously quite strictly regulated. So you'd need to consider accreditation, shareability of results, etc. However, and we did have some conversations around that as well, that doesn't necessarily mean that there can't be benefits in changing the statutory testing. It just means that there needs to be a lot more work and thought around ex ante determining potential benefits and to make sure that the ultimate outcomes are comparable or better. And by that, I don't necessarily mean just the pure test performance. I mean the overall outcome of the testing. So for example, if portable tests are performing less well, but you can do a hell of a lot more, that might still overall be preferable. So we're currently exploring also how that could look like for formal import testing at ports. And in terms of the non-statutory testing, sort of a number of different scenarios were highlighted. Some of those relate to customer assurance. For example, norovirus and shellfish tests would be really useful to prove safety to consumers. There's also an opportunity for improved quick hygiene testing. So, for example, environmental swabs of countertops where speed is obviously key. So, you know, immediately if you need to re-clean or also for process trials in factories. And another scenario was to help with production decisions. So, for example, test irrigation water to know which water sources is or isn't safe or aren't safe to use. And yeah, then the specific test requirements obviously depend very heavily on which scenario of those that we are looking at. So, for example, test performance in a number of cases, or actually, I suppose in a lot of cases, what you really want to know is whether there are any viable pathogens present. So anything that's infectious and can actually cause an issue rather than just pure absence presence. Also, for example, for ubiquitous pathogens, often you'd want to count on a threshold, whereas if you're interested in a particular strain, you'll obviously, that might be a very bad one where you just want to know if it's present or absent. And another thing that came up quite a lot across the board is the need for a portable test to not throw up a lot of presumptive results, so which would require retesting, which would then obviously counteract your potential time savings. The cost question is always an important one, and actually very often for additional testing, even if there is an obvious advantage of having faster results, costs would still, or we were told costs would still need to be either comparative or comparable or cheaper than the current lab standard. However, there may be scenarios where the investment could be made by a specific group or organization, for example, agronomists, who could then offer a service to anyone who wants the testing done. And then lastly, the ease of use. So most, or actually all stakeholders were happy with some degree of complexity. So it would just require proper training and training protocol, obviously, but that wasn't really seen as a problem. The main issue here, I think that was mentioned, was that obviously we're talking about food production environments. So if you don't have proper separate laboratory facilities, you don't really want to be messing around with live cultures of the stuff that you want to keep out. So yeah, there's quite a few opportunities and sort of various ways in which those could be prioritized, which we're still discussing the final detail of. But yeah, essentially what we want to make sure is that we use scenarios that are widely applicable, possibly transferable, but also make the most use of this being a pilot study and being able to explore possibilities. And I think with that, I will hand back to Ashley.
Okay, thank you. So after considering the results of our literature review and assessment of technologies and then the needs of stakeholders, we went on to create a short list of technologies which we thought we could take forward for the pilot study in Work Package 3. So firstly, I'm just going to briefly talk about a few promising technologies which we haven't included and why. So firstly, sort of device technologies such as lab on a chip and biosensor devices. So these may be promising future technologies in diagnostics, and a lot of research has been done into these, so they featured prominently in our literature search from the past 5 years. However, in the literature, they have not progressed far beyond the prototype stage for the in-scope pathogens. So maxing out sort of around TRL5, and we didn't feel like the scope of the pilot study could contribute to the development of these, or it wouldn't be where our efforts would be best placed, because we wouldn't want to just add another prototype to the many in the literature, which may be unlikely to progress further and commercially available for in-scope pathogens. We wouldn't be in a position to sort of validate or test a current prototype. Other technologies which were promising but which we didn't choose to take forward were nanopore sequencing, which also hasn't progressed past TR5 for the target, mainly due to all the components needed, for example, a suitable DNA extraction, suitable portable PCR platform, and nanopore sequencing device, and a way to do data analysis. Another big hurdle was the complexity of use and data interpretation for end users, and we thought it made more sense to try and progress other simpler molecular technologies in the field first, and these may act as a stepping stone for the use of nanopore sequencing in the future. So the first technology that was on our shortlist, which I want to talk about, is portable real-time PCR. So many portable real-time PCR devices are now commercially available, and the speed of PCR in the field is also rapidly decreasing. And we identified some of the platforms which had the most potential to be taken forward for foodborne pathogen detection. So the advantages of these is that they can give rapid results, so between 30 and 60 minutes, depending on the platform. And they can be combined with PCR assays, which are published and available in the literature. So the tests available could be rapidly expanded on these platforms. And they can be multiplexed to include test controls, which can give more confidence in results. And often in the literature, they've displayed equivalent analytical sensitivity and specificity to laboratory real-time PCR tests. And there are commercial kits available for in-scope pathogens on multiple real-time PCR platforms. So the disadvantages are that they will require a more complex protocol than other on-site detection technologies, including pipetting steps, with some having more liquid handling steps than others. The cost of buying the instrument could be unaffordable for some end users, and you're looking at sort of £5 to £10 a test minimum, with 10,000 to 20,000 initial investment in the instrument. So if we did take this forward, the next steps would sort of be maybe verification of the available kits in the lab with all the necessary components for on-site testing, such as DNA extraction, optimizing the performance for a certain scenario, and then demonstrating the optimized test in an in-scope setting with relevant end users. The next technology I want to talk about was LAMP. So this is another molecular technique suitable for on-site testing, but has some benefits over PCR, as the equipment needed can be much simpler. So LAMP has multiple formats, so it can be used with real-time instruments measuring fluorescence or turbidity, but it can also be paired with more simple readout methods such as LFD or colour change. Its advantages are that it again can have similar analytical sensitivity and specificity to laboratory tests such as real-time PCR, and there are assays available in the literature for all of our target pathogens. But it also can be made relatively equipment-free, which real-time PCR can't at the moment, if you use these colour change or LFD readouts. The disadvantages are that it is again a relatively complex protocol, including pipetting steps. It can have a high cost, especially if wanting to buy one of the real-time instruments, and sample-to-answer kits are less readily commercially available, although there are companies which could probably readily make the kits if there was enough interest in buying them. And then the next steps for taking forward these technologies, for using the real-time machine, we would again probably start with verification of a test with all the necessary components, and verification in the lab, and then going on to field validation with end users if they are available and willing. And then for the other formats, such as the colour change or LFD, we might test multiple formats to see which one was best, and then verify this with an in-scope pathogen in the lab, and then go on to test the performance in a relevant setting with end users. The next technology we're considering is a chemiluminescence method. So this is based on a chemiluminescence probe, which can be covalently linked to a specific enzyme liable group. Then if the target bacteria is present in the sample, it expresses this specific enzyme, which can cleave the enzyme liable group from the chemiluminescence probe, and this results in a reaction and causes an emission of light, which can be measured. So the technology is currently available for swabs of surfaces for certain bacteria. The advantages are that it's a very simple protocol with a closed system, so you've got very low chances of cross-contamination. It's a relatively cheap test with less capital investment needed, and it has high sensitivity. It detects only live bacteria as they need to be expressing the enzyme. And there are commercial kits available for in-scope pathogens. The disadvantage is that it does require an enrichment step, so it's not going to be a super rapid test, somewhere between 8 and 24 hours, depending on the pathogen. But this obviously could still provide some speed advantages over sending samples to a laboratory. The next steps we take with this technology is either verification of its diagnostic performance in an in-scope scenario, as the kits are sort of intended to be used, and then to get end user feedback and identify any specific logistical barriers to deployment. Or we could try and adapt the tests for different matrices in the lab, such as food rather than swabs, and then optimize and test their performance. So there's already been some published work optimizing the kits for dairy, so it'd probably be possible to maybe use them for other food matrices. And then lastly, we have lateral flow tests. So the advantages of these are obviously the simple protocol and interpretation of the results, no instrument needed, and a relatively low test cost, and commercial kits are available for all in-scope pathogens. So the disadvantage is that they have quite low sensitivity unless combined again with an enrichment step, which obviously makes the process more complicated, and they only produce qualitative results. But the next steps we could take with lateral flow would again be to generate performance data in an in-scope setting where LFDs are maybe not currently used but could be useful, and then assess the generated data to see whether we think they are performing well and are a useful test in that scenario, and collecting end user feedback of any barriers to deployment. So then finally, I just wanted to pair some of these technologies we are considering to scenarios which were mentioned by stakeholders as potential settings for on-site testing. So firstly, we have listeria swabs for environmental monitoring. So we thought this could be paired with LFDs or the chemiluminescence tests. So these would be used as a risk management tool for factories and food processing or production settings, with a focus on ready-to-eat products. As this would not be a required test, but an additional test some companies might want to perform, cost is likely to be a key factor, and why these technologies were chosen, and also the feedback given that generally simpler, easier to perform tests would be better. Next is E. coli as a fecal indicator and another risk management tool, which was mentioned for shellfish, meat, and also irrigation water for fresh produce. And we would pair this probably with LAMP or real-time PCR. This is because generally it was mentioned that quantitative results would be most useful as E. coli is widespread, and also fast results are key for decision making. And then finally, testing for norovirus in shellfish and water would also be paired with probably real-time PCR or LAMP. So the need for better norovirus testing in the shellfish industry was mentioned a number of times as a risk monitoring test, as there is an industry drive to prove safety to customers. Speed was key for the shellfish industry, and also the need for quantitative results for monitoring trends would be useful. So our next steps are to try and further narrow down this list to try and take forward 2 technologies for pilot study in a relevant setting. And that's everything for me. So I just wanted to acknowledge everyone who's involved in the work and also for funding the project. So thank you.
Thank you very much, Ashley and Barbara, for that really comprehensive overview of the interesting work that's been going on with Workstream 3A. So just looking over to the questions and answers section that we've got one question that's come in from David Walker. David, I'm not sure if the question got answered during the presentation, but if you wanted to come off mute and just ask the question if it's still relevant, and if anybody else has got questions, please feel free to drop them in the Q&A or raise your hand. David, are you there? We can see that you're off mute, but can't hear you at the moment. No, he's gone back onto mute. So maybe having some technical issues there. I don't know if can't see any more questions popping up in the Q&A side. Any hands raised? We'll give it a few minutes just in case anybody has anything specific. So can see Mark Hylett with a hand up.
I thank you. That's a really interesting piece of work. I'm from the I'm a meat hygiene inspector, so I can really see the value in what you're doing, as well as some of the implications you're going to get down the line. So and I know it's really early stages, but I was wondering if you've got any percentages. So if you take these mobile units into plant and operate it on meat, and you say, let's take Campylobacter on chickens, and you find a high level, what you're classing as a high level, what percentages are you looking at as a high level, in the individual bird, but in the flock? And then if you do find the high level, what's the processes ongoing there after the detention and retention of the birds, just moving forward, and all the implications that brings? But really interesting piece of work.
Yeah, thank you. I think there are obviously things that we definitely have to consider before anything was implemented, and this is just sort of a pilot study to look at sort of the feasibility of using those technologies. But yeah, I understand they definitely need to sort out the sort of implications of positive results and what they actually mean.
Rick, did you want to jump in there? Was that in response to that question, or is it a separate one?
Yeah, I mean, Candy, yeah, Mark, I mean, and this has been one of the challenges, and this is why I think, you know, as Ashley and the team said, you know, when we've been talking to people, you know, particularly industry, for example, they're quite keen on the non-statutory ones for possibly obvious reasons. But I think and so yeah, those are exactly the kind of things that if you got to a stage where you thought this has potential, this technology, this platform could work, you obviously then go into a whole load of kind of legal, statutory, policy kind of implications. How do you implement it? So there's definitely those things that need to be considered, and all those sort of things that you've talked about there are absolutely valid. So there are lots of work. I think you know, as Ashley said, this is very early stage. Do these things actually work? Could you take them into a site out away from a lab and actually get them to work? So it's, yeah, well, I mean, if we're realistic, these things are probably years away from, you know, full deployment, even if we get a good run on these things. But it is very valid. And I think, you know, your point about how we implement them and how we sort of co-design any new testing regime with people like meat hygiene inspectors is really critical. So absolutely, there would be a whole load of work to be done beyond the feasibility. But this has given us an opportunity to check out these technologies and see, you know, are they feasible, basically. Thanks.
Thanks, Rick. I can see David got your hand up again. See if we can get past the technical issues.
You hear me?
Yes, we can. Yeah.
Great. Yeah, don't worry about the other question that's been answered. Lateral flow device. So yeah, thanks. It was a really interesting overview of those technologies. Quite timely as well, I think. But one of the things that you mentioned at the towards the end there was like real-time PCR and norovirus and shellfish, which is a good example because it's that would that's going to be really challenging to do in the field, and something we've been thinking about for years. I was wondering, during your literature review, if you come across technologies for helping out with the preparation of those samples, because one of the key challenges is with norovirus in particular in shellfish is it's there at such low levels that even with a PCR, it's really hard to get enough material in the first place to be able to detect, even with a PCR based method. And then there are other challenges alongside that, but that being key. If you sort of come across anything.
Well, yeah, definitely come across the issue of like sample preparation being the biggest hurdle for norovirus testing in shellfish. And no, I can't say that I've seen it put together with a method that at the moment I would say would be suitable for on-site testing. So I think, yeah, the biggest hurdle with that one would be to get to sort of the TRL6 level where you've got a suitable sample preparation and DNA extraction combined with the portable real-time PCR. So no, that's definitely the biggest challenge we identified as well, reading the literature, that I couldn't see that had been solved yet.
Yeah, okay. Well, it's a shame that you didn't come across anything that could help. Yeah, we'll just keep thinking about it.
Thanks, David. Flipping back over to the Q&A, we've got a couple of questions that have come in from Purvi over at EA. I don't know if you want to unmute and pose the questions.
Hi, Elliot. The first question I have is about the literature review you were talking about at the first stage. So would you be elaborate more, like, what criteria or key terminologies you use for this work?
Inez, I don't know if you can jump in if you know more which the key search terms were for the literature search.
Yes, sorry. So we, as Ashley mentioned before, we split it in two. So we have like a more broad search and a more specific one for foodborne pathogens. So from the top of my head, I can remember all of them, but for the foodborne pathogen one, we have things like foodborne, point of need, infill, different ones referring to the infill diagnostics. And then we discard things that discard things like mycotoxins that we weren't looking specifically for, or allergens. And for the more broad one, we're basically focusing on infill, point of need, and more broad, like including DNA, bacteria, for example, virus. And I think those were the main ones. There might be another ones as well, but right now I can't remember. But yeah, that was it.
Yeah. Thanks, Ins. And another question I have, like, whenever, like, all the technologies you're talking about, that's in connection with either bacteria or virus, but can we use them for all other pathogen types apart from this too?
I think lots of the technologies would be transferable across pathogens, but I think it would just depend. Ones are pretty transferable to all pathogen types. But yeah, it would depend on the technology. I don't know if that answers the question.
No, that's fine. That's okay. Thanks. Thank you.
Thank you for that. Purvi, if there were specific other types you consider in there, please do feel free to kind of drop us a message and we can always follow up and consider further around the work that's going on. So flipping back over to the questions and answers. So we've got a follow-up point, I think, from Mark around cost being key, about who pays and who operates them is also key. So those factors, I think, that were touched on. So I don't think, Mark, if there's anything else to add to that one, please feel free to raise your hand. And then we've got Simon Jones has raised a question. I don't know if you want to come off mute and pose the question.
Hi everyone. I was just thinking, with the high initial cost and obviously going forward, I think the cost of the pieces of kit will come down in time. But if you're looking for a rollout pretty soon, could you foresee a team of say MHIs or OVs going around plant to plant and doing spec testing, or would it be as the rim samples are done currently, where so many per month are taken in each plant?
Rick, if you want to jump in, yeah.
Yeah, sure. Hi, Simon. Yeah, I mean, I think there's I think the idea of sort of I mean, people talk about a lab in a case kind of approach, and you could have mobile units that go around and do testing is totally feasible. And I think how you deploy the technology is really up for grabs. I think one of the scenarios you might think about is border inspection, where we're building. As we speak, we are building border inspection points. Could you create something that is a simple laboratory at those where you have some of this kit? And it goes back to some of the earlier questions that were asked about how transferable is this technology. The team here has specifically looked at foodborne pathogens, but I've often said this before, DNA is DNA. So, DNA from a bee, from a tree, from, a human, it's all DNA, and it's the same sort of chemistry, if you like, the test. You design different assays, you have to validate them, etc., all that kind of stuff. But if it's got DNA or RNA, you can test with it. So you could have a couple of platforms, maybe one of the real-time PCR platforms, maybe a LAMP platform that you could run different tests on. You could run animal health tests on it, plant health tests on it, food tests on it. So you could have a sort of like localized laboratory capability at, say, a border inspection port. I mean, I know for a fact that some of the plant health colleagues have sort of done this in the past at Heathrow Airport, where a huge amount of imports came in there. I think the other thing is everyone talks about cost, and there is always that. It's the cost of what we currently do and the cost of the new technology, and everyone does that direct comparison, and often those costs go up slightly, but it's often the benefit that you need to work in. So you need to, it's almost the cost of the outcome that you've got that's better. So have you stopped have you found more things and stopped more contamination? Could be a benefit, in which case would you pay slightly more for that? Could it also be that actually, and again, thinking about scenarios here, industry would pay for it because they get quicker results. So often we think about it in terms of cost as maybe a regulator and an inspector, but do we then think about the benefit in terms of would industry be happy to pay for a quicker result? And certainly, again, going back to a previous life and looking at plant health scenarios, if you go and put things in storage and leave them there for 48 hours while you get the lab result, the value of that commodity has gone down. So actually, what would an importer pay for a result that they got in 20 or 30 minutes as opposed to one they got in 48 hours? So actually, the benefit in terms of better regulation and trade might be significant. Now, again, who pays for that is going to be one of those big questions down the line. But there are various ways of looking at it. So yeah, and various options again. So yeah, lab in a case, lab in a van, definitely a situation we could look at, Simon.
Thank you very much. Very informative session as well. Lots of great information. Thank you.
Fantastic. Thanks, Simon and Rick. David, I can see you've got your hand back up. Did you want to come back in on another point?
Sorry, no, Rick just covered it. Thanks.
Okay, fantastic. Thank you. So looking over at the Q&A tab, we don't have any other questions that have been flagged there. Give it a few seconds just in case there's anything anybody else wants to raise in that tab, or if anyone's got any questions, please do feel free to put your hand up as well. No, oh, there we go. Mark's just come back in.
Yeah, so sorry. Just one. With this new technology, obviously we all need to embrace it and move forward with it. Are you expecting to find more with it, like Chemiluminescence? I was just thinking of what Rick said there about the cost and the outcome. if the outcome is we're finding more contamination, more Campylobacter, then industry are going to back away from this rather than embrace it, because I think we all know what industry is like. So could there be a possibility of being victims of our own success here?
I'll pick that one up. Mark, absolutely. And you know, there's this old adage in sort of surveillance and diagnostic terms: the more you look, the more you find. And the easiest way not to find anything is to not look. So I suppose it's a case of knowledge is dangerous. So if you have this thing, what do you do with it? And obviously, again, that's that massive combination of you know, how do we talk about, you know, if we say this, I mean, it's not, and again, this is another really important point for scientists. It's not for us just to find a load of data and then throw it over the fence at a load of policymakers and say, oh, you might want to deal with that. We've got to think about the implications of what we do as well, and how that might be used, and some of it could be dangerous as well. So I think what the team have done, and they're looking at some of the scenarios and some of the situations where it could be used, is, you know, do we look at things where there's a potential benefit from industry in terms of them looking at hygiene, where they're doing routine hygiene checks, and can they improve their hygiene checks? Maybe, you know, looking at, I don't know, an example, listeria in biofilms that build up in production plants, in food factories. You know, could they then improve the hygiene, which is then an obvious benefit, but one where you're sort of helping them to help themselves get better, rather than maybe ploughing straight into a really tricky statutory situation. So there might be other things where we sort of look at the technology in slightly less contentious contexts and situations, but then consider how we might then move it. We don't just say, oh, it's too difficult, let's not do it. And you said that, Mike, you said we've got to embrace it, but it's how we do that. And maybe we just need to get people more comfortable with it. And just to say, this isn't just foodborne disease. You know, all these arguments were played out during COVID as well. You know, everyone was used to the idea that people were sick, you got a test done in a lab, and the epidemiologist got all the information. So how do you then deal with that when you're sending tests to people in their homes and they get tests? And how do you deal with them, individuals having the power of that knowledge, etc.? So it gets a lot of social science in it as well, which is why Barbara and the team have been doing a bit of that kind of work as well. So it's a fascinating challenge, but it's a really good opportunity. Yeah, but lots to do.
Thanks, Rick. I can see Simon's just come in with a follow-up question. Do you want to raise that, Simon?
Hi. Yeah, just looking at when you would like to see this technology put out into plants. I presume that's where it would be going, or is there too many varying factors for you to give a time frame?
Yeah, too many. I think it's too early to say that. And in terms of plants, Simon, I'm not necessarily sure that would be the first point of call. I mean, it depends if it would fit within existing workflows, for example, which, you know, for some of the conversations I've had with MHIs, it doesn't necessarily, I mean, if you're not doing routine testing at the moment, then, you know, there's not a direct replacement, so it would be on top. However, what you might see are scenarios, for example, imports. So at the moment, there is always almost an element of, there was already an element of routine testing for certain things like salmonella. So maybe you're replacing something you already, well, not replacing, but considering whether you could do something different from what you already do. So I think your idea with meat hygiene and inspection and plants is one thing to consider, but I reckon that would be further down the line. So but given the complexity of it, it's good to start the conversation nice and early, I reckon. So
thank you.
Cheers.
Thanks both. Mark just popped a comment in as well, just around it'd be great to be kept up to date with progress in this area. So yeah, we definitely will. Kind of links back to that pinned Q&A that I've put in there, and kind of loops back around so that we've got the website, we've got these series of webinars, we've got a newsletter. If questions come up after this, we're more than happy to be approached. So we've got the pathsafe@food.gov.uk. We're here, we won't be disappearing, so yeah, we'll be sharing that information. The programme is, as a whole, is due to report out around March 2024. We'll obviously keep updates going in between, but that's the kind of target end date for the programme, and we'll be doing a lot of communications around that as we near the end of the programme. So unless there's any other immediate questions, I can't see any more coming up in the chat. Can't see any other hands raised. As I said, please feel free to reach out to us if something comes to mind later. So to thank everybody, thank the presenters for a wonderful presentation, everybody for attending. I've done the reminder plug around all of our newsletters and contacts, and also the next in the series of this webinar series will be on Thursday the 13th of July, running from 11 till 12, and looking at the first steps towards an environmental surveillance system for antimicrobial resistance in England. So please do join us for that session if you can. And with that, wish you all a very good day, and thank you for attending.
Thanks, everyone.
Thank you.
Great session.
Thank you.
Thanks, FERA.
Thank you.
Thank you. That
everyone.
was really
Nice to meet you.
interesting.
Thank you.
Have a good day.
Thanks,
Bye.
everybody. Have a good day.
Thank you.
Thank you.